In part 2 of chapter 1, we begin to look at the important experiments throughout history that not only allowed us to discover these small organisms, but allowed us to show that those organisms can lead to diseases in humans. Some examples of diseases that were so widespread and impactful that they really shaped human history are listed here. So your book talks about how the smallpox virus was brought over by European settlers. This particular virus is incredibly lethal, but it's particularly dangerous to individuals who have never experience this infection. None of the population has immunity to it, so they are all susceptible. The bubonic plague, which led to the Black Death. Oftentimes, if you've read, like historical fiction and things, you may have heard the bubonic plague reference. This is a bacterial infection that wiped out roughly a third of the European population. The influenza pandemic, this was a Spanish influenza. Remember, influenza virus is a respiratory infection. Over the course of about the span of a year, so 1918 on into 1919, killed about 50 million people. If you look at the worldwide population numbers, you can actually see a dramatic dip in the overall numbers because so many people died due to this particular influenza virus. The AIDS epidemic, while AIDS in the U.S. and other developed countries is very manageable as a chronic condition, there are some places around the world where AIDS is a really horrific epidemic that kills men, women, and children. So back in part 1 of chapter 1, we defined a microbe as an organism that is smaller than what we can see with the naked eye. So our first stop in history is looking at some of the first microscopes that were developed and allowed us to actually see that there was something that was smaller than what we could be aware of otherwise. So our first compound microscope was made by a scientist named Robert Hooke. So the magnification power of the lenses, the glass lenses that he was able to grind, had a total magnification of about 30 times, which means that with that relatively small total magnification, the specimens that he was going to be looking at were eukaryotic specimens. So the image that's depicted up there in the top left is a sketch of some mold that he was able to view with his microscope. He drew this sketch as well as a number of other sketches of his specimens in a pamphlet called Micrographia that he published and distributed to people. Robert Hooke was actually the first individual to coin the phrase cells. So when we think of cork, you might think of like a teacher's classroom and they have like a cork board where they would pin, papers, special announcements, things like that. Most of that now is synthetic cork, but natural cork is a derivative of trees. And so the cork that Hooke was looking at under his microscope was a plant-based specimen, and it had those very brick-like cubes, those cells kind of stacked and sandwiched together. So that's where that name came from. Antoine van Leeuwenhoek is another important developer of a microscope, but he was not a scientist. He was a draper. So he was in the business of manufacturing and then selling and purchasing different types of fabric. And he wanted to know the quality of the fabric so that he could make smart purchases. He developed his magnifying lenses in order to see the number of threads within the cloths that he was buying and purchasing. So he had a total different reason for developing his version of the microscope. But he was able to grind his lens the lenses that he used in a different way. We still don't know exactly how he was able to create such precision lenses, but he was able to create these precision lenses that allowed for a higher magnification. Additionally, he was able to have a focus adjustment, and you can kind of see what this looks like. It's very different than the microscopes that we looked at in chapter 3. So our lens is here. This is where you would view, and the specimen sat up on top of the sample holder. You had a focus knob, and then you could move the sample up and down so that you could look at it in multiple directions. So he had a totally different reason for making a microscope, but this was a time where it was becoming more well known that there were teeny tiny things that were in substances that you couldn't see with the naked eye. So just kind of out of curiosity, I guess, more than anything, Van Leeuwenhoek looked at pond water and was able to see moving substances, moving organisms within that pond water. He observed blood cells. He's the first one to observe sperm cells as well. So when he took a sample from his teeth, took some scrapings, he described what he referred to as small animals, or sometimes animalcules. And he noticed that they were in fewer numbers after he consumed hot coffee. So he noticed that that environment, that hot coffee, affected the microbes in his mouth. So once we establish that, yes, there are indeed things that are smaller than what our naked eye can see, we begin to wonder, gosh, how do they get there? So there was a popular theory called the theory of spontaneous generation that began to kind of make its rounds as an explanation for where these microbes were coming from. So the theory of spontaneous generation basically says that microbes are going to arise, they're going to be generated spontaneously. The conditions will be just right and they'll appear without parental organisms. So this was really pushed forward by individuals who were doing chemistry experiments. And their thought process was that the spontaneous generation of microbes was much like a precipitate forming when they were performing some of their experiments. Just when the conditions were right, they could get a precipitate to come out of solution and they could see it. And so that's where they were coming from. That was their point of view. But the church said, no, we don't support spontaneous generation. We hold to the idea that all living things, no matter how small, have to have parental organisms. And you know what? The church is the one who got it right. So this was at a time where religious figures, priests and monks, were heavily involved in a lot of science and a lot of research. So one of the first guys that we're going to talk about is Lazzaro Spallanzani, and he did not support spontaneous generation. Okay, so he was correct. The chemists were wrong. So spontaneous generation is incorrect. So Spallanzani set about trying to disprove the theory of spontaneous generation. So I've got a picture from a different textbook that I have used. Note over the couple of slides that we're going to continue through, the images from these are not in your textbook. Your textbook really comes up short on like visual depictions of these experiments. So I've pulled some from some other resources to help us visualize what is going on in these experiments. So Spallanzani sets off to disprove spontaneous generation. So here is his setup. He had two different kind of scenarios that he works for. So in the first one, he's going to begin by creating a broth using meat. So they, during this time, you can't just, from a scientific company order media. You can't just order broth. So what they did was they would boil leftovers from their own meals, and that would have the nutrients that they would need to be able to support microbial growth. So he creates this broth from meat and then boils this broth. Now, we know that he has sterilized it. He has killed off all of the living microbes within it. He has this sterilized broth, but he leaves the flask open. He then waits a couple of days and begins to see clouding of the broth, indicating that microbial growth has occurred. We've got binary fission that our bacteria are undergoing, and there are a lot more bacteria now. So we then have another twist on that scenario of events. We've got that same meat broth. We boil it, but then Spallanzani puts a stopper on the top of the flask and seals it. And so he is trying to show that there are no parental organisms in there, and we do not see spontaneous generation. So this supports his claim that spontaneous generation does not occur. Furthermore, after he opens this broth where he's seen no growth for a long time, he has this clouding over. So we now know that when that broth is left open, even if it's sterilized, eventually stuff from the air is going to settle in. Those microbes will utilize the nutrients. They'll undergo binary fission and grow, and then we'll get that clouding of the broth. So Spallanzani was really initially pleased with his experiment, but not everybody was pleased with it. We'll see what their hang-up was here in just a second. But during the process of the experiments that he did, one of the things that Spallanzani did was he was able to observe these microbes under a microscope. He had a sufficiently powerful microscope to look at bacteria. And he noticed that in a location where he initially saw one single cell, after a relatively short period of time, there were two that were very close to one another. He was the first to observe binary fission. So one parental cell splits into two. Now this type of reproduction was something that was new. But this was the first time that it was shown one single cell can actually be the parental cell, resulting into two. And then those two cells can go on to be parental cells themselves as they undergo that cell division. Okay, so that's Spallanzani's efforts and attempts to disprove spontaneous generation. But those that supported spontaneous generation weren't convinced. They believed that the fact that Spallanzani had sealed the flasks and put that stopper in the top, it didn't allow any oxygen into the flasks. This was a time where we thought all living organisms needed oxygen to live and survive. We now know that that is not the case for all microbes. Many microbes grow without oxygen. But this was a big hang-up for some of the people who were still holding on to the idea that spontaneous generation was correct. So Louis Pasteur is our next notable historic figure. You may recognize his name as part of the word pasteurization. So we'll talk more about him when we learn about pasteurization later in the semester. But Louis Pasteur kind of stumbled upon this idea of disproving spontaneous generation with the development of a piece of equipment called the swan neck flask. So that's what's depicted down here. This was actually developed as a way for Louis Pasteur to investigate why some wine and beer that was being produced was souring. So if you don't know, the way that wine and beer is normally produced, it's in what's called an anaerobic environment. That's an environment without oxygen. The organisms that metabolize the sugar and convert it to alcohol are yeast. So these are eukaryotic, single-celled yeast, fungal organisms. And in that anaerobic, oxygen-free environment, their metabolic processes produce alcohol as a byproduct. Now, if you just have yeast in your vat, you get that alcoholic beverage that you want. If your vat is contaminated with bacteria, when many bacterial species grow in that oxygen-free anaerobic environment, they don't produce alcohol. What they produce instead is acetic acid. Do you know what acetic acid is? It's vinegar. So when we have bacterial contamination of the vats, we end up with a soured drink, a vinegar drink that nobody wants to consume because it doesn't taste good. So Louis Pasteur was in the midst of trying to understand why we had this souring. We now know that it was bacterial contamination, but that's his whole purpose in designing this swan-necked flask. So what we have here is very much a similar setup to what Spallanzani had. We've got a broth down here that we're going to heat up, and we're going to sterilize it. Again, we don't use that term at this point in history, but we know that heating is going to make it so that nothing grows. But this curved swan neck flask allows, and it's a tube, it's an open glass tube, like a straw that's been bent. What it's going to do is it's going to allow oxygen in. So this helps to kind of quiet the critics that say, oh, well, you get no growth, but it's because you don't have oxygen. Well, now we have oxygen, and we're going to see that we don't get growth. Let's explore these steps. So we have our broth, and we're going to boil it again. We're going to sterilize it. We're going to kill all of the living bacteria that's in here. So this swan neck flask then is going to remain open. When we have those dust particles and those microbes that are contaminating the air coming in, they now get trapped in the bend and they settle at the bottom of the bend. They don't continue up the other side. So this broth stays sterile for years. He had some of these sitting up on shelves in his lab for years, and as long as you didn't tip them over, none of those microbes ever reached the broth, so it remained sterile. You could still show, though, that the broth was capable of supporting growth by tipping it, introducing those microbes that were in the bend of that glass tube, and then we would see growth very, very quickly. So Louis Pasteur's swan neck flask helped to further push us toward this idea that spontaneous generation is not how things work. But there was one gentleman by the name of John Tyndall who tried to replicate Pasteur's work using a swan neck flask, but was unable to. So the key here is that Tyndall was using a different type of broth. Again, at this time, we're just collecting things that are left over from dinner plates, and in this particular instance, some leftovers that his horses and other livestock didn't consume. So he used a hay broth. He picked up some hay, put it in water, boiled it to drive the nutrients out into the water, took the hay away, and then he had a hay broth. But when he completed the exact same steps that Pasteur did, he didn't tip his swan neck flask, but it still grew bacteria. It became cloudy. And so it took a very long time for us to figure out what was going on with Tyndall's experiments. We learned at a later date that Tyndall was unable to replicate Pasteur's experiments because the hay broth that he used had been contaminated with a type of bacteria that produce endospores. So recall that spores are tough, dormant structures that just a few of our gram-positive species can produce. Those species are in the genera Bacillus and Clostridium. So these are two groups of bacteria that are capable of producing spores when conditions are unfavorable. When conditions are unfavorable, say you're being boiled as the hay broth was, it will induce a process of sporulation. So sporulation is just when a spore-forming species actually forms a spore. So these really tough, resistant structures remained in that broth and were not destroyed after that time of boiling. So once the broth was cooled and conditions became favorable again, those spores reverted back to the vegetative state. Vegetative meaning that they are actively dividing and undergoing binary fission. Now they're using the nutrients in the hay broth. That was the explanation. At the time that Tyndall was doing this experiment, he could have done it 100 times and not gotten it to work at all. We now know that the only way to destroy endospores is through high heat combined with high pressure. So the structure, the piece of equipment that you see down here in the right is called an autoclave. An autoclave is the only way to reliably destroy not only living cells, but also endospores if a particular sample happens to be contaminated with those. So for those of you in lab, we have an autoclave, and just to ensure that we've destroyed all living cells and any endospores that may be present in our broth, we autoclave all of the nutrient agar and nutrient broth that is used in our lab. All right, so let's begin to make the jump from just identifying the fact that microbes exist to making the connection that they can cause disease. And this took a surprisingly long time to make the connection between microbes and the diseases that they cause. And there are several hypotheses for why this might be, but one pretty widely accepted hypothesis is just the fact that we were able to show that microbes were ubiquitous. They were everywhere. We were scraping them from our teeth and looking at them in pond water, and we had no reason to link those microbes with diseases because they weren't causing disease. So the idea of a certain type of microbe causing a particular disease, it took a whole lot of experiments to get to that point. So we now describe what is called the germ theory of disease. And it states that a specific disease is going to be caused by a specific microbe. And I realize that that sounds really simplistic and like you're going, well, duh, I mean, that's common knowledge, right? At this time, it wasn't. So I'd like to watch just a little clip from a video. Again, it's narrated by Bill Nye, one of my favorite guys. And we're going to start kind of midway through this. We'll cover the rest of the video at a later time. He probably gives a little more credit to Louis Pasteur for developing germ theory than he probably should. There's a lot of people, not just Pasteur, who performed a variety of different experiments that really supported and led us to this idea of one germ causing one disease.
Pasteur had lost 3 of his 5 children to typhoid fever, which perhaps explains why he was determined to find the cause of infectious diseases. It was Pasteur's work on behalf of the beer and wine industry that put him on the right track. Pasteur was trying to find out what was spoiling so much of the country's wine production. He discovered that the spoiled wine was contaminated by microorganisms, germs, and the germs were causing the wine to sour. But with a simple heat treatment, he showed that the germs could be killed off and the wine saved. The pasteurization process was born. So when it came to finding the cause of infections and contagious disease, Pasteur knew where to look. Germs, he said, cause specific diseases, and he proved it through a series of experiments and demonstrations that led to his great discovery, germ theory.
The germ theory literally marks the beginning of modern medicine. The germ theory has one central idea, that one microorganism causes one disease in everybody. Now today this seems so obvious, but this is one of the most revolutionary concepts in medicine.
So let's look at some of the important individuals in medical microbiology. So one of the first is Florence Nightingale. She's totally cool. If you are going into nursing, you should get her biography. But she was born into a very affluent family, and this was at a time when people who were wealthy did not become nurses. It was considered kind of below them or beneath them. But it was something that she was really passionate about, and so she worked with several other medical professionals and really kind of trained herself. Her kind of primary contribution, or maybe like when she became most well known, was during the Crimean War. So it was during this war that she began to take care of soldiers. And she noticed the cycles of infectious diseases. We didn't understand how they were passed, but she realized that these diseases were accounting for more than half of the mortality among soldiers. Now, her opinion was different than what was held by the military at the time. They believed that soldiers were getting sick because they were too stressed out or because they weren't eating good enough food. And those are certainly things that are going to play into this. But she was the one who really pushed the idea that it was a transmissible disease that was being passed among these individuals. And it wasn't just her observations. She began keeping track of the numbers and the spread of these infectious diseases throughout the individuals, that she was tasked with caring for. And so she actually ran the numbers. She was trained in statistics and a fair amount self-taught in mathematics as well. And she began to really push hard to convince the higher ups in the military that they need to change their policies in how they have soldiers who are being cared for, how they are kept, and grouping individuals that show these transmissible diseases away from the other individuals. So she enacted a lot of policies that resulted in saving the lives of many soldiers, not just within the war that she was dealing with, but it was able to spread. And she trained many nurses who went out and cared for a lot of individuals using the policies that she developed. Robert Koch is another key player in the story of medical microbiology. So he was studying cutaneous anthrax in livestock, so primarily sheep and cattle. So Bacillus anthracis it's one of our spore-forming species, although that doesn't have anything to do with this particular instance. But the spores that lead to this type of infection are commonly found in the soil. So this particular bacteria, when it gets in a cut on the skin, can lead to really severe deformations of the skin, and it can be lethal. So if you are in the business of raising animals and they are dying due to this particular bacteria, that's a big deal for you. So Robert Koch was a physician who had a lab. One of his primary contributions to medical microbiology, and again, I realize that this seems very obvious, but it's what's called the chain of infection. The chain of infection basically states that you can take the blood of a diseased animal and assume that it carries your pathogenic microbe, and you can introduce it into a healthy animal, and by doing so, you can cause the disease in that healthy animal. So again, really it's just the idea of transmitting microbes and causing diseases. So he not only looked at Bacillus anthracis, but he also studied Mycobacterium tuberculosis, which is the bacterial causative agent of tuberculosis. So we'll talk more about this chain of infection here in just a minute, but one of the things that is important in terms of Koch's contributions is that in his lab, we developed the use of a substance called agar to solidify liquid nutrient broths. So for those of you who are in lab, you may know this, but for those of you who are not, I want to explain that a Petri dish is a roughly 4-inch round plastic or glass dish with a slightly larger lid that sits loosely on top. So this Petri dish is a kind of storage container for this solidified nutrient media. Agar is a powdered substance. It is a derivative of seaweed, so like red algae is typically where it is isolated from. And to make a Petri dish with a solid agar media on it is very much like making Jell-O. You take your liquid nutrient broth, you heat it up, and then you add your powdered agar and you stir it up until that agar dissolves, and then you pour that hot liquid into the bottom of your dish. You cover it up and you let it cool, and as it cools, it solidifies. It very much has the texture of Jell-O. So this was one of the types of media that he used to culture or to grow some of the different types of bacteria that he was studying. So with the development of, development of the idea of the chain of infection and then the addition of this new type of media, he was able to create what are called Koch's postulates. Now there are 4 of these. And this essentially walks us through the details of the chain of infections. It allows us to show that a microbe causes a disease. So the 4 postulates that we're going to talk about are depicted over here on the left. So postulate number 1 is that the microbe has to be found in all individuals where they have that disease, a given set of signs and symptoms. And that microbe has to be absent from all healthy individuals. So we have our healthy rabbit up here because there's no microbe present. This rabbit down here has died, and we can isolate the presence of the bacteria. So postulate number 2 then states that that microbe isolated from that diseased host can be grown in some type of media. It may be a liquid broth. It might be a solid agar. But that media provides the nutrient to allow the bacteria to multiply. That then is introduced into a new healthy individual. So we take the microbe that we've grown on our media, we inject it and introduce it into a healthy host that didn't have that, and then we see that animal contract the disease and succumb to the disease. So it has to be the same set of signs and symptoms that are characteristic of that disease, and then we obtain that strain again. So we have shown multiple times now that that microbe causes that disease. So Koch's postulates are still really important, but we do recognize that there are some limitations. So there are times where really we have to use Koch's postulates as a guide rather than a hard kind of set of rules and steps that we're going to follow. So some exceptions to these Koch's postulates: tuberculosis in humans is actually an example where in some humans, so TB is a disease that is characterized by difficulty breathing, coughing fits. Oftentimes individuals will cough up blood. But there are some people who will have the bacteria that lives within their lungs, and it doesn't lead to the diseased state. So this is a time where we may not be able to follow all of those postulates like really closely because we were unable to follow the first postulate. when we start talking about a lack of an animal molecule, of an animal model, that can really make things difficult. In fact, there are times where we just ethically cannot work through Koch's postulates because there's no animal that can have the microbe grow within it, or it doesn't cause the disease within that animal. HIV, which is the causative agent of AIDS, is one of those examples. So with HIV, that's a virus that is strictly infective to humans. We can't infect rabbits or mice or even apes with HIV. And so it's just not ethical for us to try and fulfill Koch's postulates with that particular instance. So there are several times where we are unable to fulfill them exactly, but they're still a very important guideline even today. The last important point, I guess, in medical microbiology was the first vaccine. So an immunization is a kind of deliberate introduction of a weakened pathogen with the goal of causing a person's immune system to respond to and remember what a pathogen looks like. So we'll talk more about the immune response, but we've mentioned before that there is an adaptive component to it. There is a learning part of the immune system. Edward Jenner was the first one to make observations that led him to take advantage of this learning side of the immune system. So we credit Jenner with developing the first vaccine, the first immunization, and it was against the virus smallpox. So smallpox is it has since been eradicated, let's say that. Smallpox is a virus, though, that causes these pustules to form all over on a person's body, and from those pustules we get like pus, like white kind of fluid, thick fluid. So Jenner made the observation that there were some individuals who got a weak form of this pock-like disease called cowpox, and it was called cowpox because it was present on the udders of cattle, and the individuals who typically got it were the women who were milking the cows, so the milkmaids. They would get it typically on their hands and arms, but it wouldn't spread throughout their body, and it was virtually never deadly. Smallpox, on the other hand, was very much a deadly disease. And so Edward Jenner decided that he was going to attempt to deliberately infect an individual. He chose an 8-year-old boy to infect with cowpox with the idea that his body would learn what cowpox looks like and make a response to that cowpox. And it would learn enough what it looked like so that the immune response could then fight off or prevent smallpox should the virus be introduced. And that's what he did. So what's depicted over there is the deliberate inoculation of the boy with smallpox. He waited a period of time and then came back and put pus from smallpox into the boy in another lesion, and the boy did not develop smallpox. He was protected from that infection by being exposed previously to that weaker version. And so this kind of got around, and people became more willing to get a smallpox immunization to protect themselves. They got a cowpox immunization to protect them from smallpox. I want to make sure I say that correctly. So let's take a quick peek at an animation that shows this. Let's go to this one here.
This is the smallpox virus. It infected the pharaohs, caused a terrible impact on the Greek and Roman empires, and decimated Native American populations. Nothing seemed to stop this lethal plague until the end of the 18th century. At that time, smallpox was a major global endemic disease. Only in Europe, it killed around 400,000 people per year. It didn't differentiate between rich and poor. Even 5 kings were killed. But for some reason, milkmaids and shepherdesses seemed to be immune to the disease. In fact, they were renowned for their beauty because their skins were free of the scars smallpox caused. They were attractive and resistant. But why? That's what the rural doctor Edward Jenner was trying to understand. He knew that milkmaids got small blisters on their hands when they milked cows suffering from cowpox, a mild disease that cattle suffered from, which caused similar lesions to smallpox. And this phenomenon made Jenner think that maybe cowpox and smallpox were related, and that infection with the milder disease would be enough to protect people against the lethal one. As a man of science, he decided to prove it. And to do this, he needed 3 things. Cowpox that he extracted from a milkmaid, fresh smallpox from a patient, and a human guinea pig. He chose his gardener's son, an 8-year-old boy called James Phipps. He infected the child with cowpox, waited a couple of months, and injected him with smallpox. And James didn't become ill. He repeated the experiment with other patients with the same results. Jenner was right, even though he didn't fully understand how his remedy worked. The most remarkable thing was that his discoveries were made way before Jacob Henle proposed in 1840 that infectious diseases were caused by germs, a theory demonstrated by Robert Koch. Decades later, Louis Pasteur, inspired by Jenner's experiments, showed that attenuated microbes could be used to prevent diseases, and he proposed the terminus vaccine in his honor. Almost 200 years after Jenner's experiments, and thanks to global vaccination campaigns, in 1980, the World Health Organization announced one of humanity's biggest achievements, the complete eradication of smallpox, the most deadly infectious disease in history.
All right. So we do credit Edward Jenner with developing the first vaccine. Now, I will tell you that that is how this whole story is presented in your textbook. Just the past couple of years, there have been some medical history buffs who have done some research into this and shown that Edward Jenner was likely not working alone to develop this kind of process of vaccinating individuals from smallpox by using cowpox. So if you have heard about that in the news, or if you follow any of those kind of medical history things, this one may change over time. But this is where we're at right now, even despite the fact that there are huge ethical concerns that would surround this type of research. We would never do anything like this today. But that wraps up the second part of Chapter 1. So make sure that you read Section 1.2 and 1.3 in your textbook, and then complete those questions. If you have any questions or need any help, please let me know.